In this lecture, we'll be concluding our discussion on protein function, and we'll be discussing two examples. One is immunoglobulins, and the other are the muscle proteins actin and myosin. Okay. And this is chapter 5, section 2 and 3. And this should be, I would think this would be one of the shorter lectures of the semester. We're going to give a couple of examples here. The real detailed description of these probably not really necessary, and to get really to have an understanding of the details, reading the book is probably uh, essential. I'm going to give a little bit of a description of each of these, but to fully understand them, I think reading the material is definitely the best way, especially with the actin and myosin muscle proteins, is the best way for you to get a good understanding of what's actually going on. Okay. Before we start that, I just like to come up with a review question here about hemoglobin, and that was the subject of our last lecture. And if we look at this chart, it shows in red myoglobin. This is a oxygen binding chart. So it's showing the percentage of the sites that are bound by O2 versus a partial pressure of oxygen gas. And red is myoglobin's curve, and green is the hemoglobin that is in normal adults. Blue is the curve that you get from the hemoglobin inside of a fetus. So it's labeled as HbF, okay, fetal hemoglobin. Okay. So why do fetuses use HbF instead of HbA? Well, if we look at the curve, is the fetal hemoglobin, does it have a higher binding or a lower binding? Well, the numbers down here are the P50s, okay, and it's a lower number, which means it's binding oxygen stronger. And why do they use that? Well, if you think about it, a fetus, when it's inside the mother, it doesn't have oxygen in its lungs, it's not breathing. So it needs a stronger hemoglobin than the mother's hemoglobin to be able to take oxygen from the mother, right? And that's what this hemoglobin does. It's actually a composite. It has the alpha subunits as the normal HbA does, but the beta subunits are replaced by what's known as the gamma, hemoglobin gamma subunit. So it's slightly different, makes the overall hemoglobin tetramer bind oxygen more strongly. And that gamma subunit is expressed for the first few months of life. So there's when the baby's 4, 5, 6 months old, that is still being expressed as the most prevalent form of hemoglobin. Okay. Okay, moving on. Section 2 describes antibodies. Okay, and there's the immune system. There's a few different parts of the immune system. The cellular immune system can target the cells that are in the body, the body's own cells that have been infected. Okay. It can also target viruses, virus particles, bacteria. Okay. So invading pathogens. Okay. It's made up of things called macrophages, killer T cells, and inflammatory T cells. There's also what's known as the humoral fluid immune system. And this really targets extracellular pathogens, so pathogens that are in the sort of bloodstream or they're not within a cell, they're extracellular. Okay. It can also recognize foreign proteins, and this fluid immune system makes soluble antibodies. And those antibodies, in a sense, can keep a memory of past infections. And what's important in this fluid immune system would be things like the lymphocytes and helper T cells. Okay, so an cellular immune response, what happens, antibodies bind to fragments that are displayed on the surface of invading cells, like a bacteria cell, or on the surface of a virus. Okay, so antibodies will bind to that, and then special cells known as phagocytes recognize the invaders based on the antibodies that are attached to them, and they bind to them via the Fc receptor on the surface, okay? And then they in a sense engulf the invading virus or bacteria, and digest them in a process known as phagocytosis. And macrophages are large phagocytes that can ingest things as big as a bacteria cell. Okay, the fluid immune system, this is where infections are fought off with soluble antibodies. Okay, and they bind specifically to certain things that we refer to as antigens. Okay, so antigens are substances that stimulate production of antibodies. Okay, that they can be numerous things. Typically, they're bigger, they're macromolecular in nature, and they're recognized as being foreign by the immune system. Okay. So things like the protein shell of bacteria, or the protein capsule of a virus, that would elicit an antibody immune response. Surface carbohydrates of cells or viruses also would do the same. Okay. Antibodies are proteins that are produced by the B cells and specifically bind to their antigens. Okay. The binding marks the antigen for destruction, or sometimes it can interfere with the antigen's function. Okay. A given antibody will bind to a small region of the antigen, and that region that the antibody recognizes we call that an epitope. Okay. One antigen can have several epitopes. So if you have like a non-native, the easiest example for me to discuss this is a protein. Okay, so if you have a foreign protein that's injected into your body, that protein, it becomes an antigen, and antibodies can bind to different regions of that protein. We call those regions that different antibodies bind to, we would call those different epitopes. Okay, so what do antibodies look like? Well, this, in particular, we're talking about immunoglobulin G, okay, when we talk about antibodies in this class. And immunoglobulin G, the schematic of it is shown here, sort of a cartoon schematic. It's comprised of two heavy chains. So the heavy chains are shown here in blue, and then two light chains, which are shown here in different shades of red. Okay, and those are all linked together via disulfide bonds. Right? And they make, it sort of makes a Y shape. Okay, and that's when you see antibodies depicted in a cartoon form, they're typically in the shape of a Y, and this sort of explains why that is. Okay. There are conserved domains or constant domains, which are labeled with a C, and variable domains that are labeled with a V. Okay, so each light chain has a variable domain and a constant domain. Okay, the V sub L means it's variable light, and C sub L means constant light. Okay. The heavy chains have one variable domain and 3 constant domains. Right. The variable domains are all up here at the sort of Y end of this antibody. Okay. And in between, sort of in this cleft between the light chain and heavy chain, that's where the antigen binding sites are. And the variable domains make up the different antigen binding sites. And they're hypervariable, which means that there are many combinations of these variable domains, and that's what gives this specificity to a large number of different antigens. This is a crystal structure showing the approximate shape, Y shape. Okay, the red would be the light chain. Okay, and these would be the variable domains, and the rest of this would be the constant domains. Okay. Antigen binding causes very significant structural changes to the antibody. Okay, so this would be the conformation if we used a space filling model and shaded the different types of amino acids different colors. Okay, this would be the antibody conformation when no antigen is bound, and when an antigen binds, this is the conformation. So you see that there's the position of the colors here is changing. So there's a pretty significant change there. And this is, showing this picture, but showing the antigen actually in that pocket there in red. Okay, so this is following that induced fit model that we talked about in the last lecture, where the substrate or ligand comes in in this case we call it an antigen, okay, that is binding in our protein in the binding site, and it's causing a structural change to our protein. Okay. Antibody specificity is actually really important in the lab, in biochemistry labs, and we can use it in many different analytical techniques. In specifically something known as a Western blot, and also an ELISA. And we'll talk about what an ELISA is on the next slide. Okay. In a Western blot, what you would do is you would run a typical protein gel, like an SDS page gel, where you're separating the proteins out based on their molecular weight. And then you take that gel and you transfer the proteins from the gel into a paper membrane. Okay, and the proteins get stuck in the paper membrane. So you can think of this gray area down here as a membrane, and the proteins get stuck to the surface of that membrane. Okay. Then what you do is all these other sites that don't have proteins on them, you block them with a non-specific protein. And how you do that is you soak your membrane in milk. Okay, so the milk proteins will stick to any open slot in your membrane. Okay. Then you put what's known as a primary antibody on your membrane. You soak it in a solution of primary antibody, and those primary antibodies, okay, here shown as our Y shape. Okay, so this is our variable domains. Okay, they recognize specific antigens. So if you have a protein of interest and you want to look at protein X in a certain tissue sample or a certain cell sample, you would run your SDS page, transfer it to your membrane, and then put your antibody that's specific for that one protein in your solution and incubate. Okay, so that will bind very selectively to the protein you're interested in. Okay, that in itself wouldn't be very helpful, but then you incubate with what's known as the secondary antibody. Okay, and that antibody recognizes the primary antibody. Okay, so this antibody, in a sense, it's an antibody that binds to other antibodies. Okay, so it'll bind, and then the other interesting thing about it is it's attached to an enzyme on the other end of it, and that enzyme you can use to detect its presence. Typically, it's something like horseradish peroxidase, where you can use a reaction that gives off a chemiluminescence, that gives off light, and you can visualize how much of a protein is there based on how much light is being given off. Okay, so that's kind of the Western blot picture of this. The same technique is used in what's known as ELISA, which stands for enzyme-linked immunosorbent assay. And ELISA, you're doing a similar thing with a very specific antibody for your protein of interest, and then it gives off a certain color of light, and you can read that in a plate reader, so you can look at a number of different samples at the same time and see, okay, which one of these has, you can quantitate how much protein of interest is in each sample. And so these techniques are very specific, and because of that, they're very widely used in biochemical laboratories. Okay. And moving on to the third section, and this section discusses muscle protein and muscle structure. Okay, so muscle fibers, they're made of very large, elongated cells that have multiple nuclei in the cell. So it's a very kind of a strange cell. They're formed when many different progenitor cells come together and sort of fuse together. Okay, but I guess it's not your typical cell that when you think of like a cell, a picture of a cell, it's not what you would typically think of. Okay, each muscle fiber contains about 1,000 what are known as myofibrils. Okay, and myofibril, if we look at in this description here, they're these little tubes. Okay, and they're if you look closer, you can see that they're more fibrous. Okay, each one of these myofibrils is comprised of many different fibers. Okay, and there's these alternating light and dark patches and lines. Okay, so two that we're going to talk about. One is the I band. Okay, the I band is where you have light patches and a dark ring in the middle of that, which we call the Z disc. Okay, and then the other is the A band. Okay, that's where we have the dark patches, and then the line in the middle of that A band is called the M line. Okay, I don't know offhand why They're called I band, A band, Z disc, M line. Maybe the book talks a little bit more about that. I didn't get that from reading it, but it's something that it'd be good to remember, and it at least I, off the top of my head, don't have a good way of remembering that. Something that you kind of just have to memorize, I guess. Okay. The I band and A band together make up what's known as the sarcomere. Okay. These myofibrils, what they're made up of are thick filaments of myosin. And myosin is a muscle protein, one of the muscle proteins that we'll be talking about. Okay. Myosin comprised of two myosin proteins will connect together through this supercoiled alpha helix. So each of these different shaded colors here is an alpha helix, and they're coiled around. The alpha helices are coiled around each other. Okay, and then at the amino terminal part of myosin, it's a globular protein structure. Okay, so these would be the heavy chains here are shown in red of myosin, and there's other light chain proteins that are shown here in blue that attach to the globular part. Okay. So the head groups are 17 nanometers, and the tail is about 150 nanometers, just to give you an idea of the scale. Okay. And in myosin itself, the myofibril, many different myosin proteins kind of connect together facing both directions and make a filament structure, okay, of many myosin dimers connected together. Okay, the other muscle protein is actin. Okay, and actin also makes filaments. Okay, and so actin, what we see here is the little spheres that you see. That would be known as G-actin, which you can think of as standing for globular actin. Okay, they're the individual actin globular proteins, but they stick together and form a strand that's sort of helical. Okay, and there's actually another strand that interwraps with it. Okay, so both of these are kind of follow the actin and myosin follow this sort of super helical structure. Okay, and this fibril or filament of actin is referred to as F-actin. Right, the F you can think of as standing for filament. Here's a space-filling model of this, just to show you. There's the actin filament. The lighter shade is one chain of actin. The darker shade is another. Right, they're wrapping around each other, and then the myosin head group can fit in this sort of a channel that's made by the actin. Here's a little bit better depiction of what's actually happening here. So in a typical, shouldn't say typical, in an uncontracted muscle, which is shown here on the top picture, okay, you'll have your actin thin filaments, okay, and your myosin thick filament, and there's sort of very little overlap. Right, so this region of the thick filament of myosin where there's no actin overlap, that would be the A band. Okay, so this corresponds to the A band, the darker region. Okay, in that that M line that we see, you can, I guess, now that I'm thinking about it, you can think of the M line as standing for myosin line. And that's when the termini, I guess, since it's plural, of these individual actin fibrils come together. Okay, so then they're facing from that M line. They're facing opposite directions. Okay, but the C termini line up on the M line. Okay, so that's relaxed muscle. Okay, the I band, the lighter region here, that corresponds to these actin thin filaments. Okay, and then the Z disc is this structure here where they're connected to. So this is only showing from here to here. Okay, but there'd be another other actin filaments going this way and another myosin filament there in the center, okay, on this side and on that side. Okay. When the muscle contracts, what happens is the myosin head groups walk along the actin filaments. So the thick filament myosin walk along the actin thin filaments. And you can think of as moving these actin filament structures closer together. Okay. And that's what we see here. So the dark region right here kind of goes away, and the lighter regions kind of get smaller as well. Okay, so the Z discs are coming closer to the M line. Okay, Z disc is coming closer to the M line. Okay, so how does that happen on a molecular level? And that's what we'll try to explain here. And in this process, it takes energy, chemical energy, and that is in the form of ATP, adenosine triphosphate. Okay, and this ATP, the energy used from ATP, causes conformational changes in a protein, and in particular, it's the myosin protein, and that's required for the function. Okay. So this is the figure your book gives. Okay, and it's important to note that this is a cycle. So there's no real beginning and end to this, but the book kind of picks this point as the start. Okay, and then you go through a cycle, right? If you look in, there's probably more complete descriptions of this if you look online, but some of the figures you see online either start at a different spot or maybe have a slightly different number of steps to this. So just be aware of that, I guess, if you're looking online for a more detailed description. It might not be 100% transferable to this description, but this is the one we're going to use. Okay. So at step 1, in this step, we have a myosin head group that is connected to the actin filament here, and it's connected at this. They color these a little bit differently so you can see relative motion. So it's connected at this dark orange actin protein in the actin filament. Okay, ATP binds to the myosin head, and this causes dissociation from actin. So binding ATP will make that myosin head group dissociate. Okay, so now we have an ATP bound to our myosin head group. Okay, the ATP is then hydrolyzed. Okay, so it is converted from ATP, so adenosine triphosphate, to ADP, which is adenosine diphosphate, and a phosphate group. And in biochemistry, we depict phosphate groups by themselves as this P sub I, and that I stands for inorganic phosphate. Okay, it's a phosphate group that's not attached to an organic molecule. Okay. So we have ADP and inorganic phosphate associated with that myosin head, okay, and it's dissociated from actin, not bound to actin like in this form. Okay. And it had a conformational change by hydrolyzing that ATP. The energy from that was transferred, and it moved that myosin head group. Okay. The myosin head group then attaches to the actin filament, okay, in a different spot now. So now it's at that, I guess, lighter orange colored actin molecule in the filament. And it connects there, and it releases the inorganic phosphate. Okay. The release of the inorganic phosphate triggers the power stroke conformational change in the myosin. So the myosin head group, it makes what's known as a power stroke, and that moves the actin and myosin filaments relative to one another. Okay, and ADP is released in that process. Okay, and so then it starts all over again. Okay, so now, so after we've gone through the one cycle, now we're not attached to the dark orange anymore. We're attached to that actin molecule next to it, but this cycle can be repeated. Okay, and you can see by doing that cycle, you're going to use a very large amount of ATP. This depiction is just another one I found off the internet that was I thought was pretty good. It gives a little bit more description than the one in our book because it talks about why calcium's important here, and these other proteins called troponin and tropomyosin. Okay, those are also very important, and they're part of the actin thin filament as well. Okay. So I included this as another sort of description that might be a little bit easier for you to read through. Okay. All right, so it's actually pretty important that the myosin binding sites on actin they are regulated by these proteins, okay, troponin and tropomyosin. Okay. Their regulation avoids, continuous muscle contraction, and a continuous muscle contraction is something like a cramp. Okay? So when you get have problems with electrolyte levels such as calcium, you can get cramps, and this is one of the reasons why. Okay? Nerve impulses will release calcium, and calcium causes conformational changes to this tropomyosin troponin complex. Okay, and those changes that binding calcium cause, it exposes the sites that myosin bind to. Okay, so that's why muscle contractions require calcium, and they're controlled. Right, we can control them. Right, you can control when you want to bend your arm because the nerve impulses can let out calcium and allow for the muscle contractions that you need to perform fine motor movements. Okay. Right, so those are the descriptions of protein function for immunoglobulins and muscle proteins. Okay, next time we're going to be talking about enzymes, and will be our first introduction to enzymes. And really what an enzyme is a protein that catalyzes a reaction. I know I've said that before, but you'll probably hear it again. Okay. and we'll be giving an intro to enzymes and then discussing how ways in which they work.